My focus will be a case study of a sexual population. And to explain, I see this forward. Okay, so the outline of my talk will, first I will talk about the situation in Sweden, and then we will see which variation is present on the barberry, and I will say something about the outbreak about the of stem rust and wheat in 2007, and then I want to mention something about the difference in the epidemiology between clonal and sexual populations. So this is something that was advertised to farmers in Sweden in the 50s and 60s, and it's the life cycle. I think many of the previous speakers have introduced my talk in a very, very good way, so I don't need to talk about the different spore stages. Eugene did a splendid job just a while ago. But of course, to be able to fight the disease, the main method to do that was to eradicate the barberry, because then you took away the overwintering possibility in Sweden. And there was a law of barberry eradication similar to in UK. It was in force from 1918 until 1994, and since 1994 the number of barberry plants have increased in the country. The map here is showing a screenshot from the Swedish like the people interested in flora and fauna. They go into this webpage and note down the GPS coordinates, sometimes a picture, where they have found a species. And this is the record of the barberry species. And the species in black, they are the ones that are susceptible to stem rust. And the species in red are the ones that are resistant. As you see, the most common species is Berberis vulgaris, which is the common European. And then the second one is Berberis thunbergi, which is this ornamental plant. But as you know from previous talk, these ones can, many of the crosses are also present, and many of those are also susceptible to stem rust. So to understand the epidemiology of stem rust in Sweden, we have to understand the climate and what happens. And as you know, it is winter, and it's cold, and nothing is growing during many months. But in early May, everything starts sprouted, and the barberry leaves are coming out. And the first symptoms of cluster cup rust, which stem rust is called on barberry, is usually in very last days of May or early June. And then you can find this aecial until mid-July, end of July. And then in the first symptoms of stem rust on oats is usually found in end of June or the beginning of July. But the really heavy infections is usually found after mid-July, and that is when the crop is after heading and flowering. And that is also when all the advisors go on vacation. So usually they don't see the stem rust infection. So it's not a big problem because they don't see it. But if I go there in 20th of July, it's usually full with stem rust. In oats. Yes, and then it's also in maybe the first weeks of August you start to see the teliospores, and then a couple of weeks later the crop is harvested. If we would have talked about the yellow rust, which is not have a sexual population in Sweden, you would have seen the first pustules in the field probably around early June. So that's quite different. So in my thesis, I looked at the variation using microsatellite markers on the different hosts. So if we look at this, this big cloud here, That's the samples collected from barberry. And then here in the little corner, you have samples collected from rye. And in this corner, you have samples collected from oats. And so you see there is a big diversity on barberry. It's not only samples that are individuals that may infect oats or rye in this case. It's also a lot of other grasses. And many of us know that it's more than 360, or it's about 365 species that have been reported as a host to stem rust or Puccinia graminis. So we were interested in the diversity on barberry and like the detailed diversity. And from my colleagues' work on the pine blister rust, they showed that each aecium is a single individual. So I wanted to test if it's the same for stem rust and crown rust. So we collected acia, and then we cut out these letters represent each single acial cup or aecium, and the group of aecium is acia. I cut out the single horns, and then we genotyped them using microsatellite markers. And as you can see, there are, within one group, there is a variation in one of the alleles. So we think that there is one pycnia that is infected by different pycniospores from a different source, and that creates a large diversity also already on the barberry bush. And our Danish colleagues found the same thing. Julian published his paper about Striformis, and they find the same thing, that there's a very large variation within each acia. So what about this 2017 outbreak? I was called in early September last year, and they have found something that they didn't recognize. And I think this is remarkable because in previous times, stem rust was very well known, but it was such a long time ago, so people don't recognize it anymore. And on the right, you have a picture from the field, and actually everything you see is teleospores. It's not secondary infection of anything, it's teleospores. It was a lot. I've looked at a lot of rust, but I've never seen this heavy. It was also on the ears, and it was bad. So of course I took samples from that field. I also tried to see if I found symptoms in the surrounding fields, and I found in winter wheat, I found in other spring wheat, I found on barley, I found on some wild grasses within the area. And I also remembered, because I used a trial about 20 kilometers away from this place, and in that location, I had a month earlier, I had seen some very, very weak stem rust infection, but nothing unusual. But then I went back, and then I found some symptoms on winter wheat there as well. And of course, my colleagues all got a little bit interested in this, and they were also serving in other parts of the countries, and this was quite localized. No other incidents was found. So, as you saw in the previous picture, it was very much teleo stage, which means that you have to go through the barberry to produce uridinospores that can be tested for pathotyping. So I took every single brown spore I could find, and that is basically the ones on the picture. It's both from some wheat and some barley. And then Mogens and his colleagues kindly accepted to help me, and Mechan has done the initial testing of the pathotyping. And then he wanted to know if he could exclude some clones. And so I genotyped about 64 samples or 69 samples that he sent me, and this is the tree that come out of it. So from these few plants, we got the same number of individuals. There were we didn't find any clones, and of course this was repeated to make sure that it was really the true result. So how does this outbreak connect to, sorry, we have to talk about Meron's result as well. So what was done was that he took 10 bulk samples of this sexual population and inoculated on the 20 differentials. And here you see the results. You can see where they were, that there are some SR24, the absent virulence is SR24, SR25, 31, and 38. And the rare ones, which are in light, they were only found a few pustules, but they were present in the population. Yes, so Meron is continuing working on this, and we hope to be able to update you on a later stage. So if we compare this to the previous outbreak, this outbreak is here in the yellow, and the one with the rings around is the samples from winter wheat, and then you have some barley crosses behind. So you can see that these ones clearly belongs to the same population as the rye, but they are a little bit different. They group in one section. And if we connect this to known races, this is a Bruford distance tree showing the population. And you have the blue ones, they are the live samples from Meron. The green ones are the ones I collected as teleospores in the field. And the yellow ones are the references that kindly were provided both by Mogens and Annemarie and Les. And I think you should note that this, in the middle here between the green one and the first yellow one, is a very thin line, and that means that it's a very large distance between the samples, a genotypic distance. So it seems that and then we have one of the references, I don't know if you see it, but the big yellow arrow will show that the samples collected in Sweden clusters together with this SCCLS clade number 4, that Les calls it. And that is this 7A that has been sequenced, the reference genome. So it means that it's a quite old North American with possible background in Europe and sexual, yeah. So it's something that's so it doesn't match with the UG99 or the recent German or recent outbreak in Italy. So what does this say? The sexual we know what we know about the sexual population. We know that they are more likely to form teleospores. We also know that virulences are conditioned by recessive genes. That means that if you have a virulence in the population, it's quite likely that it will continue to stay there. We also know that the timing with the host is, and the phenology is crucial for disease development. So in a sexual population, on the x-axis you have the number of virulences within an individual, and then you have the number of offspring. So we know that it's a normal distribution with offsprings. So most of them will be they will not cause a problem. But we have a few that is in this corner that will potentially cause a problem, and those are the ones that we should think about. So just to illustrate the difference between a clonal rust infection and a sexual rust infection. In a clonal, you have a few spots 1 week, and then maybe the other day you have another infection in another part, and then they after a couple of weeks they will start to produce offsprings in the close proximity, and then they will spread, and then maybe another one will come in, and then at the end you have the whole field infected. But if you have a sexual population, you remember that each acial horn will produce a new individual. Then you have, at one day, you will have one population deposited on the field. And the next day you will have a different population deposit on the field. The third day you might have a new one, and the fifth, and so forth. So, and then they will of course produce offspring within the field. But in the case of Sweden, where you have this forced sexual population, I I think the very rapid from a very clean field to a very infected field, it must be due to this massive spore deposition. So to sum up this, we think that it's a forced sexual population. It has to go through the sexual cycle. And we really think that the alternate host is a part of the Puccinia graminis life cycle in Sweden. We know that we have very many different races, and we also think that it must be established within the area. And the thing that this was a very late sown crop, and it was a very cold summer that delayed the maturity of the crop. That's probably why we got this very massive infection in this particular field. But I think it would be very interesting to follow the epidemics for next year. And that's all. Before, I want to thank Anders Lindgren at the Swedish Board of Agriculture, who I work with in doing the surveys. And then especially Jonathan Ewan, who was the one who initiated the barberry work, being my PhD supervisor. Maria Jonsson for doing the lab work, and Les for providing references. And then, of course, my funders. Thank you.